Welcome to Biotanemy. Sequencing studies. Sequencing is the process of determining the precise order of nucleotides in a DNA or RNA, or the sequence of amino acids in a protein. Over the years, many sequencing techniques have been developed. Let's discuss some of the important ones in the coming videos. In this video, we'll discuss about Maxim-Gilbert sequencing technique and Sanger sequencing. Maxim-Gilbert sequencing. Maxim-Gilbert sequencing is a method of DNA sequencing that relies on the chemical cleavage of DNA to determine the sequence of nucleotides. It is developed by Alan Maxim and Walter Gilbert in the 1970s. In this method, the DNA is first labeled with a fluorescent marker or radioactive isotope phosphorus at 5' The sequence is then denatured into single strands. The single-stranded DNA is now treated with different chemical reagents that selectively cleave the DNA at specific bases, such as DMS or dimethyl sulfate cleaves at adenine nucleotide. The hydrazine will cleave at cytosine and thymine, and the addition of sodium chloride to the hydrazine will make them to cleave specifically at cytosine. And addition of formic acid will cut at adenine and guanine. The modified DNA bases are then cleaved by hot piperidine. The fragmented nucleotide is then separated using agarose gel electrophoresis. Finally, the gel will undergo autoradiography to visualize the separated DNA fragments. Advantages: 1. Purified DNA can be read directly. 2. Homopolymeric DNA runs can be sequenced as efficiently as heterogeneous DNA sequences. 3. It can be used to analyze DNA-protein interactions, that is, DNA footprinting. 4. Analyze nucleic acid structure and epigenetic modifications to DNA. Disadvantages: 1. It requires extensive use of hazardous chemicals. 2. It has a relatively complex setup. 3. It is difficult to scale up and cannot be used to analyze more than 500 base pairs. 4. The read length decreases from incomplete cleavage reactions. 5. It is difficult to make Maxim-Gilbert sequencing-based DNA kits. Sanger sequencing. Sanger sequencing is one of the important sequencing techniques that have been widely used by scientists until now. It has a low error rate and is 99.9% accurate. The Sanger sequencing is also known as the chain termination method. It makes use of the PCR technique and works by selective incorporation of dideoxynucleotides or DDNTPs to halt the DNA synthesis. DDNTPs are similar to DNTPs, but lack one hydroxyl group at its 3' carbon of the sugar, deoxyribose. During Sanger sequencing, a DNA primer complementary to the template DNA is used to be the starting point for the DNA synthesis. For nucleotides, adenine, thymine, guanine, and cytosine are given as deoxynucleotide triphosphates or DNTPs. Also, the polymerase is added to extend the primer. The 4 DDNTPs of adenine, thymine, guanine, and cytosine, labeled with 4 distinct fluorescent dyes, are also added to the mixture. During sequencing, the DDNTPs will be incorporated randomly into the growing DNA chain, halting the sequencing and producing DNA of different lengths. The synthesized DNA is then denatured to single strand, and then separated by sides with the help of agarose gel electrophoresis. The sequence of the nucleotide or protein is determined by reading the color of DDNTPs present in the DNA fragment length order, from shortest to longest. The visualization of Sanger sequencing can be done in 2 ways, manually and automated way. In the manual method, the user will read all 4 lanes of the gel at once, moving from bottom to top. While in automated Sanger sequencing, the computer reads each band in a capillary gel electrophoresis. Advantage: chain termination methods have greatly simplified DNA sequencing. Limitations: 1. Nonspecific binding of the primer to the DNA will affect the accurate readout of the DNA sequence. 2. Secondary structures present in DNA will affect the fidelity of the sequence. That's all for today's video. Thank you for listening. Hope all of you understand the concept, and for more queries and proposal, please comment down. See you all in a new topic.